terms of disease bioinformatics. It's something called Malacards. And Noah Rappaport-Mardix, a postdoc in the lab who does that, is sitting here in the front row. There are many disease databases. You see a few of them symbolically shown, and we mine many of them, and we generate a database which has about 17,000 entries with links and information gleaned automatically from some of these sources. This is the web page, and you can learn more at the poster number 8003 tomorrow from Noam. I will also say one word about CAFA, Critical Assessment of Functional Annotation. This is for Ido Friedberg, who asked me to show this slide, and it's very relevant, as you can see in the text in the middle. Genes with known function are but a drop in the ocean. A drug target for Alzheimer's may well now be marked as unknown function. So if we know more about proteins and genes, we'll know more about diseases. And with that, I will introduce our first speaker, Dr. Warren Kibbe from Northwestern University, who will give the first talk.
Great. Thank you very much. It's great to be here. Es ist sehr schön in Berlin wieder zu sein. I'll echo Lauren's comments. If people can come down a little closer, it would be, I think it'll actually make for a better set of questions at the end of each of our presentations. So what I'd like to do is go through and talk about some work that we've been doing, and actually it's been a big collaborative. There have been a lot of folks in the community looking at coupling disease ontology with human phenotype ontology and what we can do for mining with that information. And I'll talk briefly about something that my group in particular has been doing using disease ontology to mine medical records. And hopefully that will make good sense for all of you. So what I'm going to briefly do is go through what is disease ontology. I was happy to see that disease ontology was a large one of the bubbles for malady cards, and go through a brief description of human phenotype ontology, which really is work that Peter Robinson here at Charité in Berlin has been pushing forward, and then talk about Phenomizer, which is something again that Peter's been developing along with Chris Mungall, Susie Lewis, Monty Westerfield, and I think a number of other people, again, in the community have been developing. And then talk about how we can use that to link phenotype, disease, genes, and then last, like I said, I'll go into using disease ontology to mine medical records. So I did notice there was a rather large typo in the abstract. Disease ontology has more than 8,000, not 800 diseases, and I apologize for not catching that a little earlier. So the disease ontology itself is really about harmonizing our description of disease and make it easier to be able to couple information from various sources to the concept of disease. This is what the disease ontology looks like if you go to the Open Biomedical Ontology's webpage and go to the record for disease ontology, and you can see that Lynn Schrimmel, one of my partners in disease ontology, is the contact person, and she'd be very happy to take any of your comments that you may have after my talk, as am I. So I'm going to give you a brief history of disease ontology. So this is something that the Rex Chisholm and I started at Northwestern now almost 10 years ago. And our original use case was, how can we identify patients with a given disease if we look at just data that's present in their medical record? So that's something that's actually taken us nearly 10 years to get to actually use disease ontology for. But we've had a number of interesting journeys along the way. One of them was when we started, we tried using ICD as the framework for disease ontology. So ICD is the International Classification of Disease. And I have to say it was an utter failure to try to use ICD as a backbone for an ontology because it's just not organized appropriately. So we rethought all that and we tried it again, and again we failed. So this is an example where the third time, again, finally we got it more or less right, and we actually used UMLS concepts, organized them into a rational framework for disease, and I think we now have something that is much more sensible and computationally tractable. And along the way, and as you can see on the right-hand side of this, we've picked up many, many great partners out in the community. We have many more than I can list, but certainly the Neuroscience Information Framework, RGD, which is the rat genome database, Array Express, another one of the disease ontologies, so the infectious disease ontology, the cell line ontology. We've ended up with lots of groups that we've had great partnerships with to make disease ontology a better thing. And most recently working with the human phenotype ontology. So there's a recent paper that you can find in Nucleic Acids Research highlighting the disease ontology. And as I said, as of the point when this paper was published, there were 8,029 diseases listed in disease ontology. And you can go to our website. This is a snapshot of a specific disease in disease ontology. And you can look and all of the XREFs listed for a given disease entry are all hyperlinked and take you off to the proper source. So in the interest of going quickly through this, I won't go through all the classifications. But there are a number of resources. I'm going to show another one, which is the immune epitope database. And they've been very good at incorporating disease ontology terms into everything that has a disease component. And that's, again, been what we've been seeing with more and more of the partners out in the community is when they have disease concepts that they need to refer to, they're linking off to disease ontology terms. So this is a view of part of the hierarchy of disease ontology. I won't go through all the text. I'm hoping that this will get posted somewhere and all of you can download it. But if you Google disease ontology, the browser that is diseaseontology.org is number one, and I think number two is the OBO version of disease ontology. So what happens when you actually start to look at a given branch in disease ontology? So here we're looking at diseases of metabolism. You can then look at an inherited metabolic disorder. So this starts pulling us in closer to the OMIM view of diseases, so an inherited disease. And then you can look at the children of that. In this case, we're going to look at bilirubin inherited diseases and two specific syndromes. And you'll notice with both of them, there's an OMIM term that's listed. And so a little bit later, I'll go through and show exactly what those OMIM terms are and how to walk between OMIM and HPO and then disease ontology. So one of the things we think is very important in disease ontology is having as many external references as we can find to existing common vocabularies. So here we list a number of the vocabularies that are out there: OMIM, SNOMED CT, the NCI thesaurus, MESH, and ICD-9. And so for a given disease that's in disease ontology, if those terms are present in those vocabularies, we try to link to them. And again, we think that enables the community to be able to walk between these different things. At the same time, when we're talking about a specific term in disease ontology, we want to have a rich definition of what that disease actually is. And so we've been developing more and more logical definitions. We currently have about 1,000 terms in disease ontology that have logical definitions, and we're pulling terms from these different ontologies or controlled vocabularies. And I won't go into that in more detail, but I'm happy to take questions about it. And because of that richness, we've been able to use it in a number of data mining papers, and here's a few of them. But more importantly, once a month, we update a database of all the gene riff connections that list specific diseases, and that's all released at hdoa.nubic.northwestern.edu, and people are free to download that and take advantage of it. So taking the data that's actually at that website, you can do very interesting modeling and visualization pieces like this particular graph here, where it's a lot of the genes that are connected to specific diseases. And you can see it's gene networks, so that a given gene is actually connected to multiple diseases. I'm going to switch now from talking about the disease ontology to talking about the human phenotype ontology. So this is, again, something that Peter Robinson here at Charité was founded and has been the major motivating force for. And it's based on taking phenotype descriptions out of OMIM. That's really how the human phenotype ontology started out. So the vast majority of the phenotypes that are currently listed in human phenotype ontology are based on OMIM descriptors. And in fact, I think Alan's going to talk about disease ontology, and maybe he'll mention some of the phenotype pieces that are in OMIM, right after me. So the idea of the human phenotype ontology then is to build up connections between OMIM diseases, and we can reach from an OMIM disease back into disease ontology, and then also look across different phenotypes and say what diseases are involved in this phenotype, and of course what genes are involved in that phenotype. So Peter's group, along with a lot of other folks, have developed a tool called Phenomizer, and again that's linked. You can go to that link by just typing in Phenomizer and the human phenotype ontology in Google and go right to it. And you can see then what you get when you go to Phenomizer. And this is the publication of Phenomizer in a recent publication. So what happens when you actually want to do one of these crosswalks between disease ontology, and here I'm going to take a particular symptom. Again, this is bilirubinemia called Dubin-Johnson syndrome, and it has an OMIM term. So this is the disease ontology view. You can see if you go into that page on disease ontology, there's a lot of information there. There are additional common vocabularies that mention this particular syndrome. There's a MESH term for it. There's an NCI thesaurus term for it. There's a SNOMED term for it. There's the UMLS QE, and of course the OMIM term. So you can walk from disease ontology into OMIM, and here's the first bit of a very detailed clinical description of what the Dubin-Johnson syndrome is. And then you can move from that into Phenomizer. So you can look up that particular term and its OMIM ID, and you can see all of the phenotypes that have been associated with that particular disease. And since its core phenotype is that the patients have hyperbilirubinemia, you can look in in HPO for other diseases that are linked to bilirubinemia. And you can see, in fact, there are a lot of different inherited diseases that all share this one phenotype. So you can get a sense that in fact, even though a given syndrome is from a single gene, there are actually a number of genes, and that you'd expect them all to be involved in bilirubin metabolism. So quickly you can get a sense that there's a network, even just moving in this very simple sense, or this very simple example, from disease ontology to OMIM to the human phenotype ontology. and that you can imagine then that as you look at any given set of either a set of diseases, a set of phenotypes, or a term in OMAM, that in fact there's a complicated network that you can discover. So I'm going to switch from talking about that phenotype side of things to talking very briefly about some work we've been doing to use disease ontology to mine data from a medical record. And I'm going to take as my example one particular aspect of diabetes, and that's patients who've had diabetic retinopathy. So it turns out with eMERGE, so eMERGE is a large consortium in the United States where we're looking at building up detailed algorithms for identifying specific disease, that this is one of the specific disease terms that have been developed by the eMERGE consortium. So at least when I was first thinking about things like diabetes and what we would find in the medical record, that seemed like a very easy problem. But it turns out to be very, very difficult to actually identify properly in the medical record. So for diabetic retinopathy, we want to exclude, of course, all patients who are not diabetics because their source of retinopathy is different, and of course we need to look for patients with retinopathy. So there's a fair amount of work that went into just determining who in the medical record has type 2 diabetes. And part of that, the trick to that is you're not looking at blood values that are that have elevated glucose levels. You're actually looking for medications that indicate that patient is a diabetic and that their glucose is under control because of their medications. Thanks. So there's a long description of what diabetic retinopathy actually means from a coding standpoint, and again, I'm happy to go through this, but it's fairly complicated to look for. And you also need to be able to exclude a number of terms. So looking at the problem list in the medical record, you need to be able to exclude various things that say, in fact, the reason for this diagnosis is there was no evidence of this particular diagnosis. So you can be fooled by seeing codes that in fact that are exclusively calling out diabetic retinopathy for cases where that was an exclusion. And they need to have had an eye exam within the past 2 years. So that all it's a very complicated algorithm, and what we wanted to be able to do is represent something that was much simpler in disease ontology and be able to reason on that. So here's the important ICD-9 codes that are included for diabetic retinopathy. And then we've loaded that into a fairly highly used software program called I2B2, which sits on top of a data warehouse. It's a way to do analytics into a data warehouse. And I know it's a little bit hard for folks in the back to see, but when you select diabetic retinopathy and you see various subterms, you can actually run I2B2 and in a matter of seconds get back a result looking over about 2 million patient records that in this case we had roughly 2,200 patients at Northwestern with this particular disease that really, when we've gone back and done manual chart abstraction, we're at about a 95% confidence level that these folks indeed had diabetic retinopathy. So that was a way to dramatically shortcut a lot of the algorithm development that we had to go through for eMERGE. So as I mentioned, this has been a huge team effort, both developing the disease ontology itself and then all of the phenotype pieces, and then also the gene disease and phenotype connections. The folks on the left are either in Lynn's group or my own. The folks in the middle are primarily people who've been willing to provide guidance and work directly with us. And the people on the right side have been the folks out in the community that have been willing to partner with us. And I apologize for anyone whose name I've left out, because there have been lots and lots of people out there that have helped us do this. So with that, I'll say thank you and take any questions.
Please use the microphones in the aisles to ask questions.
Can I ask, or is there a line? Hi, so Warren, on your left. Hi, great talk, thanks. Could you expand a little bit, because most human disease research, at least, is not done that much in humans, but rather in model animals.
Right.
Could you expand a little bit on how you are dealing with common model animals, say mouse, rat, fly, that are used for human disease research, C. elegans, and how the human disease ontology, if at all, works, interfaces with those?
Sure. So I think there's a couple answers there. Is one, that we're still working with a number of those model organism communities to get them to use the disease ontology to represent the disease aspect of their models. The rat genome database has actually been using disease ontology IDs for their diseases for a while. The other side of it, though, is looking at the data that gets produced from those models. For instance, Array Express has been very good about taking all the gene expression data sets that get deposited with them, and when they're coming from specific kinds of models that represent that disease, they actually do use the disease ontology IDs to cross-reference then that particular disease. So again, it becomes a discoverable network of connections.
Thank you.
Warren, I think one of the challenges is to have a complete, acceptable list of diseases. And whoever does disease bioinformatics sees the challenge, and it's very difficult to meet the challenge. Could you comment on that, please?
Sure, it is a big challenge. So for instance, something that we've been, I guess I would say, challenged to do is to try to bring in rare diseases, orphanet. There's a large list of rare diseases out there. In fact, they're roughly the same size as what's currently in disease ontology. And the reason I say that's a challenge is that those groups are great. They have a lot of good disease information, but we want it to be in the same framework as disease ontology currently uses, and that requires typically a fair amount of manual curation to go through and put it in the right right framework. And there's also a fair amount of disambiguation that goes on as you're doing that, because we found that when, for instance, for disease ontology, we're still debating if you have two different genes for a given disease that are, for instance, two different omium terms, but the etiology of the disease is essentially identical. Is that one disease in disease ontology, or should it be two? So it is a challenge to come up with that single list, and I think differentiating what each of those diseases are in a way that becomes computationally tractable is also a challenge, because they start, the nuances involved can be quite subtle. And again, if you're trying to do things like natural language processing of different data sources using disease ontology, you want to make sure that you're accurate with it, so that the descriptors that distinguish the diseases are truly distinguishing them. So it's not a trivial problem, as you pointed out.